Hello, we are part of the SRM Multidisciplinary Project Initiative of the 24-25 batch, and we are group number 6. Our project is titled Determining Novel EGFR Inhibitors Using Deep Learning Based Approaches. So, what are EGFRs or epidermal growth factor receptors? Well, they are proteins found on the surface of many cells all across our body, and it plays a crucial role in regulating cell growth, cell division, and cell survival. Under normal conditions, EGFR activates only in response to specific signals helping maintain a healthy tissue function. However, mutations in the EGFR gene can disrupt this balance. These mutations often cause the receptor to remain constantly active even without external signals. And this leads to an uncontrollable cell division, which is a key driver in development of cancers. EGFR mutations are most commonly found in non-small cell lung cancers or NSCLC for short. This makes up about 85% of all lung cancer cases, and in India, lung cancer is among the top 5 leading cancers, which where NSCLC is the most prevalent subtype. Globally, lung cancer accounts for around 1.8 million deaths per year, making it the leading cause of cancer-related deaths worldwide. To combat this, targeted therapies known as EGFR inhibitors have been developed, and these drugs were initially effective. But over time, many patients develop resistance due to secondary mutations, which as a result leads to the cancer returning in a more aggressive form, creating a major treatment challenge. Adding to this complexity is the traditional drug discovery process, which is incredibly slow and expensive. On average, it takes about 10 to 15 years and up to 2 or 3 billion dollars to bring a new drug to market, and nearly 90% of potential drugs fail during clinical trials. That's why our project proposes an alternate solution to this. Using deep learning techniques to identify novel EGFR inhibitors by training models on chemical and biological data, we aim to predict a new compound that could be more effective, faster to discover, and potentially overcome resistances seen with existing therapies.
Let's now talk about the technical flow of our project. We first started out by extracting molecular data from Campbell database, including smile strings and IC50 values. The smile strings and IC50 values indicate the effectiveness of a compound as an inhibitor with 9,000 samples. After getting the data, we trained multiple deep learning models such as CNN, GNN, MLP, RNN, DTM, GAN, and BM. These models predict the PIC50 value, which measures the inhibitor potential. We faced an issue here with the values of the models because they were not ideal. To tackle this issue, we filtered out the data by focusing on highly active compounds with IC50 values below 10 nanomolar. We had it below 10 nanomolar to have biological relevance. After filtering out the data, the sample size reduced from 9,000 samples to 1,300 samples.
Then we rebuilt the models, and among them CNN and GNN had the best performance. So we built an ensemble model improving the prediction accuracy. After training and validating the model, we made a user-friendly web application using Flask. In this website, when you enter a Campbell ID, it gives the effectiveness of the molecule against the EGFR and also gives the predictable dockability as high, medium or low. In summary, our work can bridge molecular biology and deep learning to accelerate drug discovery and also help researchers to find new and potent treatments for EGFR related cancers. Thank you.